Chapter 4. Cellular Processes. Cytology. Part 2. Imagine a factory that produces not one, but thousands of different products. It operates hundreds of assembly lines, 24/7, at blazing speeds with never a breakdown. Some of its raw materials are shipped in from outside sources, but the factory also recycles some of its used substances into new ones. Now imagine that this highly organized production facility is so tiny that 100 of them could easily fit on the period at the end of this sentence. You have just imagined a typical cell, the unit that God designed to be the building block as well as the manufacturer of all living things. 4. A. Cellular energy. Although cells depend upon many conditions in the environment, a constant supply of energy is essential for them to perform all of their cellular processes. Even in the most favorable environments, cells need energy to maintain their homeostasis. They use energy to manufacture needed substances and to tear down others. In fact, when a cell stops using energy, it is dead. Expenditure of energy is a primary attribute of life. 4.1 Objectives. Explain why energy is required for life. Define autotroph and heterotroph. Describe how energy is stored in ATP molecules. Key terms: autotroph, heterotroph, ATP, adenosine triphosphate, ADP, adenosine diphosphate. 4.1 Energy relationships. Cells use amino acids and other substances again and again in the process of making many large and different molecules. Energy, however, is a one-time commodity. Every time it is used, some escapes and becomes unusable. Cells must use energy to make energy-storing molecules. Since some energy escapes during every energy transaction, more energy is needed to build an energy-storing molecule than is stored in the molecule. To make any large molecule, cells need large quantities of energy. The stored energy in large molecules is of value to the cell primarily when it is being used. Every time a large molecule is broken apart by the cell to get this stored energy, some of the energy escapes. As a result, the amount of usable energy in an energy storage molecule is considerably less than the total amount of energy in the molecule. Cells constantly battle to maintain a supply of energy. Energy, crucial for all life, is the one commodity that is stored, but not without considerable loss, is used, but never reused, and escapes constantly. Yet all living cells need energy at all times. Autotrophs and heterotrophs. Organisms can be classified into two groups according to how they obtain their energy. Organisms that make their own food are called autotrophs. Plants, algae, and some other organisms are able to capture light energy and produce their own food. They are the primary autotrophs. Autotrophs are also called producers. Organisms that depend on other organisms for their energy are called heterotrophs, or consumers. These include humans, animals, fungi, and most bacteria. Photosynthesis, the process used by most autotrophs in storing energy, making their food, is discussed in the next subsection. Cellular respiration, the process all organisms, including autotrophs, use to release energy from storage, is discussed in subsection 4.3. ATP, the energy currency in cells. When a person wants something from a vending machine but finds only a $20 bill in his pocket, he cannot get what he wants. He has enough money for the item that he wants, but he cannot get it because he does not have the amount in a usable form. Similarly, a cell may have many units of stored energy in starch or lipid molecules, but there is too much energy in these molecules to be used all at once. If all the energy in a molecule of starch or lipid were released in a cell at one time, that area of the cell would be destroyed. The energy from these molecules must be converted into smaller, usable units. In all known living organisms, from bacteria to humans, that smaller usable unit is the energy stored in a molecule of ATP, adenosine triphosphate, DN, acene, triphosphate. It could be said that ATP is the coin of cellular energy currency. Examining its structure will explain how energy is made available in ATP. The discussion of nucleic acid structure in Chapter 2 introduces all three of the ATP molecule components. The backbone of the ATP molecule is adenosine, which is made of a simple sugar and adenine, one of the bases found in DNA. The other component of ATP is a chain of 3 phosphate groups attached to the adenosine backbone. The key to how ATP stores energy is found in the bonds between the 3 phosphate groups. They are bound to each other by unstable, high-energy covalent bonds. When the bonds are broken, a useful amount of energy is released, an exothermic reaction, and is immediately available for use in any cellular function that requires energy, an endothermic reaction. Occasionally, 2 or more ATP molecules supply the energy for a single reaction. Active transport, biosynthesis of molecules, cellular movement, and locomotion are all processes powered by energy released from ATP molecules. In most reactions, it is the phosphate group on the end of the chain that is broken off. This reaction produces a molecule of ADP, adenosine diphosphate, ATP-1 phosphate group, a free phosphate group, and energy. The reaction can be summarized as follows: ATP arrow, ADP plus P plus energy. An ATP molecule that has given off its energy can be reused. With the proper enzymes and an adequate supply of energy, the ADP and a phosphate can recombine to form ATP. The reaction can be summarized as follows: a DP plus P plus energy arrow, ATP. Muscles use energy from ATP in order to contract. However, ATP cannot be brought to muscles through the blood. Why? Since the ATP molecule is very unstable, the flow of blood would break ATP down to ADP long before it reached the muscles. Thus, ATP is a temporary, unstable energy storage molecule and must be constantly manufactured by every cell as long as it is alive. A normal adult male uses about 2,800 kilocalories, or 2,800 calories, of energy each day, requiring about 150 kilograms, 328 pounds, of ATP each day. However, there is only about 0.005 kilograms, 0.175 ounces, of ATP contained in a person's body at any given time. If a person is to stay alive, his body must constantly make ATP from ADP and energy. God has graciously designed cells to be astonishingly efficient at recycling ADP back to ATP. In a working muscle cell, 10 million ATP molecules are used and regenerated every second. In 60 seconds, the same cell will have regenerated all of its ATP. This rapid recycling ensures that the cell has all of the energy it needs to function at optimal levels. We truly are fearfully and wonderfully made. Psalm 139:14, dashed down to the molecular level. 4.2. Objectives. Write a simple equation for photosynthesis. Explain how light energy is captured for use in photosynthesis. Describe the light-dependent phase and the Calvin cycle. List the final products of photosynthesis. Explain how the environment affects photosynthesis. Key terms: photosynthesis, chlorophyll, pigment, light-dependent phase, electron transport chain, Calvin cycle, stroma, ATP synthase, chemosynthesis, 4.2 photosynthesis. On the Earth, the sun is ultimately the source of energy for virtually all living things. One of the things the sun does is heat the Earth. But warmth, though essential, is not the energy needed to keep living things alive. Chemical energy is required for the reactions that maintain life. Since the sun's energy does not come as chemical energy, it must be changed into a usable form. There are certain organisms that are capable of absorbing light energy and converting it into stored chemical energy in a process called photosynthesis. Green plants and algae, the primary photosynthetic organisms, perform this essential energy transformation not only for themselves, but also in large enough quantities to supply stored chemical energy for almost every living thing. Because it is the essential step between solar energy and life, photosynthesis is one of the most important biological processes. Photosynthesis is important for another reason. It produces oxygen. Human beings, as well as animals, plants, and most other organisms, require oxygen in order to live. The following study explains how oxygen and stored chemical energy are produced by photosynthesis. Chlorophyll and light. Chlorophyll, K-L-O-R-A-Phyll, a green pigment, is the primary catalyst of photosynthesis. Pigments are special light-absorbing molecules. The chlorophyll molecule has a ring-shaped head portion containing a single magnesium atom and a tail containing a carbon chain. Although the chlorophyll molecule does not contain iron, it is necessary for chlorophyll's formation. In fact, chlorophyll is made only when the cell has a supply of iron and is exposed to light. Plants grown in reduced light or in iron-poor soil lack chlorophyll and appear pale yellow or whitish. The two primary pigments of photosynthesis, chlorophyll A and chlorophyll B, are the main types of chloroplasts in photosynthetically active cells. The chlorophyll molecule is located in the membranes of the grana. The A form of chlorophyll is a primary catalyst and serves as the primary pigment of photosynthesis. The two forms of chlorophyll differ in the wavelengths of light that activate them. Chlorophyll absorbs wavelengths of all the colors of light. When something appears red, it is actually reflecting red light waves and absorbing all others. This fact explains which wavelengths or colors of light are necessary for photosynthesis. The green color of most photosynthetic organs of plants indicates that wavelengths other than in the green range are being absorbed. The process of photosynthesis. Photosynthesis takes place in the grana of the thylakoids of the chloroplast. Chlorophyll and other pigment molecules are located in clusters of 100 to 200 in the thylakoid membrane. Experiments have revealed that photosynthesis actually occurs in two separate yet closely related phases: the light-dependent phase and the Calvin cycle. The light-dependent phase. The first phase is called the light-dependent phase, or light reactions. As the name implies, light energy is required. Light energy is absorbed by various pigments and is used to energize electrons in a chlorophyll, a molecule. The energized electrons leave the chlorophyll, a molecule, and pass through a series of protein molecules called the electron transport chain, which is embedded in the thylakoid membrane. As the excited electrons pass through the electron transport chain, some of their energy is used to form ATP. In order to replace the energized electrons that left the chlorophyll, a molecule, photolysis occurs. Photolysis is the splitting of a water molecule into hydrogen ions, electrons, and oxygen. The electrons replace those lost when chlorophyll became energized. The hydrogen ions remain in the thylakoid, and the oxygen is eventually released into the atmosphere or used by the cell. After an electron has passed through the electron transport chain, it is still highly energized. To capture the remaining energy, the electrons bind in pairs to a special electron carrier molecule, NADP+, nicotinamide adenine dinucleotide phosphate. When NADP+ picks up these two electrons and a hydrogen ion H+, it becomes NADPH. This molecule stores the energy for later use. In short, the light-dependent phase uses light energy to form NADPH and ATP. It also produces oxygen gas. The Calvin cycle. The Calvin cycle, or light-independent phase, is the second phase and takes place in the stroma of the chloroplast, where the necessary enzymes and other reactants are located. Although light is not required, the Calvin cycle depends on the products of the light-dependent phase, ATP and NADPH, and carbon dioxide, CO2, from the atmosphere. The Calvin cycle is sometimes called the carbon fixation phase, because during this phase, carbon is fixed or assimilated in an organic compound. To start this phase, a molecule of carbon dioxide diffuses from the cytoplasm of the cell into the stroma of the chloroplast, where it binds to a 5-carbon sugar called ribulose bisphosphate, RUBP. Through a series of reactions, the energy stored in the ATP and NADPH that was produced during the light-dependent phase is used to produce a 3-carbon sugar, phosphoglyceraldehyde, or PGAL. Some of the PGAL molecules are used to form glucose, while others reform RUBP so the Calvin cycle can continue. To summarize, the Calvin cycle produces sugar using the atoms from CO2, energy from ATP, and the electrons and hydrogen ions provided by NADPH. The steps of photosynthesis. The clusters of chlorophyll molecules are called photosystems. There are two types of photosystems: photosystem 1, P1, and photosystem 2, P2, based on the geometrical arrangement of the chlorophyll molecules. As with many biological processes, photosynthesis can most easily be studied in a stepwise fashion. The light-dependent phase. Step 1. Light energy is collected by the various pigments and transferred to a chlorophyll molecule, where electrons are excited in photosystem 2. At the same time, an enzyme splits a water molecule into electrons, oxygen, and hydrogen ions in a process called photolysis. The electrons from the water molecule replace those lost by photosystem 2, the hydrogen ions stay inside the thylakoid, and the plant eventually releases the oxygen to the atmosphere. Step 2. An excited electron leaves the chlorophyll, a molecule, and is picked up by an electron carrier molecule, which is the beginning of a series of molecules called the electron transport chain. Step 3. As the electron passes through the transport chain, some of the energy is used to pump hydrogen ions across the thylakoid membrane. Step 4. After the electron has passed through the transport chain, it is accepted by a molecule of chlorophyll in photosystem 1. Simultaneously, light energy excites the newly arrived electron. Step 5. The excited electron in photosystem 1 is passed to an electron carrier in a different electron transport chain. Step 6. This electron transport chain carries the electron to the stroma side of the thylakoid membrane, where it combines with a hydrogen ion and NADP+, an electron carrier molecule, to form NADPH. Step 7. The hydrogen ions produced by photolysis and those pumped into the thylakoid during step 3 begin to form a large concentration gradient between the inside of the thylakoid and the stroma of the chloroplast. Step 8. As the hydrogen ion concentration builds up inside the thylakoid, the ions pass from a higher concentration to a lower concentration through an enzyme, ATP synthase, very similar to the process of osmosis. This movement of hydrogen ions causes the enzyme's gradient to create chemiosmosis. As the hydrogen ions pass through the ATP synthase molecule, it converts the potential energy of the H+ concentration gradient into chemical energy as it combines ADP with a phosphate group to produce ATP. The chemical reaction that summarizes the light-dependent reactions is 12H 2O plus 12N ADP plus 18. ADP plus 18P plus light energy arrow 6O2 plus 12N ADPH plus 18 ATP. It should be remembered that the overall reaction for photosynthesis is 6H2O plus 6C, O2 plus light energy arrow, C 6H12, O6 plus 6O2. A comparison between the products of the light reactions and the products in the overall chemical equation for photosynthesis shows similarities. The oxygen produced in the light-dependent reactions is the same oxygen that is seen in the overall photosynthesis reaction. The ATP and NADP produced in the light-dependent reactions are used in the Calvin cycle. The Calvin cycle. Step 1. A carbon dioxide molecule diffuses from the cell's cytoplasm into the stroma of the chloroplast and is fixed or attached to RUBP, ribulose bisphosphate. RuBP is a 5-carbon sugar with 2 phosphate heads at the ends. The resulting 6-carbon sugar is very unstable and breaks apart to form 2 molecules of PGA, phosphoglycerate. Step 2. PGA is converted into another 3-carbon molecule called PGAL, phosphoglyceraldehyde. PGAL is energized by the energy from a phosphate bond in an ATP molecule and then receives hydrogen and electrons from the carrier molecule NADPH. The NADP plus and ADP released during the formation of PGAL may be used again in the light-dependent reactions to synthesize additional ATP and NADPH. Step 3. Some of the molecules of PGAL never make it to step 3. They are used in the synthesis of other organic molecules such as glucose. However, in step 3, many of the PGAL molecules are converted into RUBP, utilizing the energy from a molecule of ATP to recharge the cycle. The RUBP returns to the start of the Calvin cycle. Photosynthesis recap. Photosynthesis is divided into two major phases, the light-dependent phase and the Calvin cycle, and each of these proceeds in a stepwise fashion. The PGAL produced at the end of the Calvin cycle is used as the building block of organic compounds such as glucose. How much energy is needed to produce one molecule of PGAL? Every spin of the Calvin cycle overview of photosynthesis. Light energy, water, and carbon dioxide enter the chloroplast, where the light-dependent phase and Calvin cycle work together, using ATP, ADP, and the electron carrier molecules. Oxygen and PGAL, which is used to make sugars, are the final products. Fixes one CO2 molecule, and since PGAL has 3 carbon atoms, it takes 3 spins to produce one molecule of PGAL. 2 molecules of ATP and 2 of NADPH are used in step 2, and 1 ATP molecule is used in step 3, using a total of 3 ATP and 2 NADPH molecules per spin. Therefore, the total energy requirement for producing one PGAL molecule is 9 molecules of ATP and 6 of NADPH. The overall chemical equation for photosynthesis is 6 carbon dioxide plus 6 water plus light energy. Chlorophyll, C, 6H12, O6 plus 6 oxygen. Glucose is not produced in the actual photosynthesis reactions. It is the molecules of PGAL that are used to make glucose and other organic molecules. Conditions for photosynthesis. Adequate quantities of the right wavelengths and intensity of light are necessary for photosynthesis to occur. If they do not absorb enough energy, chlorophyll A molecules cannot become sufficiently energized. On the other hand, the rate of photosynthesis will reach a plateau and will go no faster if all of the chlorophyll A molecules are functioning at maximum capacity. The cell must also be able to absorb sufficient carbon dioxide. This is seldom a problem since carbon dioxide normally accounts for about 0.03% of the atmosphere by volume. Just as with light intensity, the rate of photosynthesis will reach a plateau when all of the chlorophyll and molecules are at maximum capacity. Proper temperatures for photosynthesis vary from plant to plant. For most plants, room temperature, 21 degrees Celsius or 70 degrees Fahrenheit, is about right. However, above 32 degrees Celsius, 90 degrees Fahrenheit, many plant enzymes do not function properly for photosynthesis. At midday during the hot summer months, many plants do not carry on photosynthesis. The lower temperature limit for photosynthesis is near freezing for some plants, but others require more moderate temperatures. In most plants, the functioning of chlorophyll A is temperature dependent. A greenhouse with improper ventilation or with drastic temperature changes can affect the supply of carbon dioxide, and photosynthesis may cease. Occasionally, a lack of water will cause photosynthesis to cease. On hot, dry summer days, the roots of a plant may not be able to absorb sufficient water and therefore cannot transport enough water up the stem to maintain photosynthesis in the leaves. Chemosynthesis. Other autotrophs. There are a few bacteria capable of obtaining energy from inorganic chemicals that they break apart in a process called chemosynthesis. Chemosynthesis. These bacteria can use that energy to synthesize sugar. Since chemosynthetic bacteria do not depend on other organisms for their food, they are autotrophs. Chemosynthetic bacteria often grow in dark areas with little organic matter in their environment. Some of these bacteria change ammonia to nitrites and nitrites to nitrates. Others use iron compounds. Probably the best-known chemosynthetic bacteria use sulfur compounds and produce sulfur dioxide. A walk through a swamp may disturb some collections of these gases that are trapped underwater. The rotten egg odor of sulfur dioxide that bubbles to the surface may have been produced by chemosynthetic bacteria. The deep-sea hydrothermal vent shown in Figure 2-16, page 46, also harbours chemosynthetic bacteria. These tiny organisms are the basis of a whole food chain that exists in total darkness. 4.3. Objectives. Differentiate between aerobic and anaerobic respiration. Summarize the major events in glycolysis, the Krebs cycle, and the electron transport chain. Compare and contrast alcoholic and lactic acid fermentation. Key terms: cellular respiration, aerobic, anaerobic, glycolysis, Krebs cycle. Acetyl coenzyme A, fermentation. 4.3. Cellular respiration. Respiration is commonly thought of as breathing, air being taken into and forced out of the lungs. Breathing is only the first step in the body's respiration. Oxygen obtained by breathing is transported from the lungs to the cells where cellular respiration takes place. Cellular respiration is the breaking down of a food substance into usable cellular energy in the form of ATP. In most cells, glucose is the primary food source of cellular energy. However, lipids, other monosaccharides, and even proteins can be used. Cellular respiration may be aerobic, requiring oxygen, or anaerobic, not requiring oxygen. Most cells carry on aerobic respiration. Many cells that normally perform aerobic respiration can, if necessary, operate anaerobically. There are some bacteria and fungi that carry on only anaerobic respiration. The primary processes of aerobic cellular respiration are discussed first, and then two important anaerobic respiration processes are presented. Comparing aerobic respiration and photosynthesis. Aerobic cellular respiration can be compared to burning. If a lighted match is held to a sugar cube, the match will supply the activation energy necessary to set the sugar on fire. The burning sugar releases large amounts of thermal and light energy. Carbon dioxide and water vapor are also released, although they cannot be seen. If the flame were put in an airtight chamber, the fire would soon be extinguished because of a lack of oxygen. In aerobic cellular respiration, the sugar glucose requires a small amount of activation energy to begin a series of enzyme-controlled chemical reactions that release the energy contained in ATP molecules. Just as the burning sugar cube did, cellular respiration releases carbon dioxide and water molecules. If cells do not receive oxygen, they cannot carry on aerobic respiration, and in time, many will die. Aerobic cellular respiration is the opposite of photosynthesis. Photosynthesis combines water, carbon dioxide, and light energy to form glucose. Aerobic cellular respiration breaks down glucose to form water, carbon dioxide, and energy. The reactants that are needed for one are the products of the other. Photosynthesis can be generalized thus: 6H2O plus 6CO, 2 plus light energy arrow. Open parenthesis chlorophyll, close parenthesis arrow. C6H12O, 6 plus 6, O2. Cellular respiration can be generalized thus: C6H12, O6 plus 6, O2. Arrow 6H 2O plus 6 CO 2 plus A, TP energy. These processes, however, are not the exact reverse of each other. For example, chlorophyll is not used in cellular respiration. Although some chemicals function in both processes, they proceed down different enzyme paths. These differences are necessary because the purposes of the two processes are different. Photosynthesis captures light energy and converts it to stored chemical energy. Respiration takes stored chemical energy and converts it to a ready-to-use chemical energy carrier, ATP. The light energy harnessed by a chlorophyll molecule during photosynthesis is made useful to all cellular processes by cellular respiration. The atoms of oxygen, carbon, and hydrogen are essentially the same after those processes. But the energy involved has been changed several times, and much of it has been lost. The process of cellular respiration. Cellular respiration may follow several different pathways, depending on the organism and the environment. Each process is discussed in steps to show the logical progression of the reactions. All types of cellular respiration begin with a common process: glycolysis. Glycolysis. Glycolysis, glyKHLUSES, involves the breakdown of glucose into pyruvic acid, hydrogen ions, and electrons. It takes place in the cytoplasm, which contains the enzymes necessary for this series of reactions. Pyruvic acid can then be used in the different forms of cellular respiration. Because glycolysis does not require oxygen, it is considered anaerobic. The byproducts of glycolysis are shuttled into the mitochondria to continue cellular respiration. Step 1. 2 molecules of ATP supply the activation energy necessary to start glycolysis. Using the energy and 2 phosphates from the molecules of ATP, glucose is broken down into 2 molecules of PGAL. Note in Figure 4-10 that each molecule of PGAL contains 3 carbons and 1 phosphate. This is the same compound seen in photosynthesis. Step 2. Each molecule of PGAL releases electrons and hydrogen ions to an electron carrier molecule, NAD+. Each PGAL molecule also has a phosphate attached to it, forming 3 carbon compounds, each now with 2 phosphates attached. Step 3. Each phosphate group added in steps 1 and 2 is removed and added to a molecule of ADP, forming a total of 4 ATP molecules. The reaction in this step produces 2 molecules of pyruvic acid. This pyruvic acid has the same chemical energy that was found in the original glucose molecule. The total energy recorded output of glycolysis is 4 molecules of ATP. However, 2 ATP molecules were needed to initiate the process. The net energy production is 2 molecules of ATP. In addition, 2 NADH molecules have been produced. What happens next depends on the type of cell and whether oxygen is present. Aerobic respiration. Aerobic respiration may be divided into 2 phases: the Krebs cycle, or citric acid cycle, and the electron transport chain. In eukaryotic cells, both of these phases take place in the mitochondria. In prokaryotes, they occur in the cell's cytoplasm. The Krebs cycle. The pyruvic acid molecules from glycolysis diffuses across the mitochondrial membrane into the matrix of the mitochondria. In the Krebs cycle, the pyruvic acid reacts with an enzyme that removes a carbon from the pyruvic acid to produce acetyl coenzyme A, or acetyl-CoA, CO2, hydrogen ions, ATP, and electrons. The CO2 diffuses out of the mitochondria and will eventually leave the cell. The hydrogen ions and electrons will combine with electron carrier molecules to be used in the second phase of aerobic respiration. These hydrogen and electron carrier molecules are carrying high-energy electrons that are essential to the next phase of aerobic respiration. Electron transport chain. The majority of the ATP energy is produced in the electron transport chain. Chapter 3 explains that the mitochondria have enfoldings of the inner membrane called cristae. The molecules that form the electron transport chain are located in this membrane. The production of energy by the electron transport chain in mitochondria is similar to the workings of the electron transport chain in the chloroplast. During this very productive phase of aerobic respiration, the high-energy electrons from the electron carrier molecules, produced by the Krebs cycle, are used to generate huge amounts of ATP, the energy coinage of respiration. In a cascade of reactions that shows remarkable design, each pair of electrons moves through the steps of the electron transport chain and, on average, gives off enough energy to convert 3 molecules of ADP to ATP. In total, 1 molecule of glucose yields 32 molecules of ATP just from the electron transport chain. Aerobic respiration energy output. Looking at the entire process from glycolysis to the electron transport chain, What is the final energy production in terms of ATP molecules from one molecule of glucose? Glycolysis had a net production of 2 molecules of ATP, and the Krebs cycle also produced 2 ATP molecules. The electron transport chain is the big producer of ATP. It used the electron carrier molecules to produce 32 molecules of ATP. Table 4-1 shows the maximum results from one glucose molecule, 36 molecules of ATP. The aerobic cellular respiration of glucose converts approximately 40% of the energy contained in a glucose molecule into the form of ATP molecules. Although a 60% loss of energy may seem wasteful, this breakdown of sugar is one of the most efficient energy processes known. In most man-made machines, cars for example, only a small amount, about 20%, of the energy available in the fuel is used. Most of the rest radiates as heat and is unusable. Anaerobic respiration. In the cellular respiration just described, oxygen is a key reactant. However, some cells exist in environments where oxygen is not available. Many bacteria in the lower layers of swamps, lakes and oceans do not have any oxygen available in their environment, and some organisms may actually be destroyed by exposure to free oxygen. Other cells that normally operate with a supply of oxygen can occasionally function without it. For example, when a person engages in strenuous physical activity, he may begin to breathe heavily but still be unable to supply enough oxygen for his muscles to function properly. Without enough oxygen, his muscles cannot carry on sufficient aerobic cellular respiration to supply the needed ATP. If cells in these circumstances are to supply themselves with usable cellular energy, they must rely on anaerobic respiration. Fermentation is the breakdown of food, usually glucose, without oxygen. There are two main pathways of fermentation, depending on how the cell modifies pyruvic acid. Why pyruvic acid and not glucose? The answer is that glycolysis is the starting point for both aerobic and anaerobic respiration. One glucose molecule is broken down into 2 pyruvic acid molecules. Depending upon the organism and the enzymes available, the pyruvic acid is usually converted to either an alcohol or lactic acid. Alcoholic fermentation. In alcoholic fermentation, the pyruvic acid molecule is changed to a molecule of ethyl alcohol in a 2-step process. Step 1. The pyruvic acid gives off a carbon dioxide molecule, converting it into a 2-carbon compound. Step 2. The 2-carbon compound takes the hydrogen ions and electrons from the carrier molecule NADH to make ethyl alcohol. The use of the hydrogen and the electrons frees the carrier molecule so that it can be used again in glycolysis. This process occurs in certain bacteria and yeasts and is used to make alcoholic beverages. Yeasts are also used in breads. The carbon dioxide production causes the dough to rise, and baking evaporates the alcohol from the bread dough. Lactic acid fermentation. This process is used by some microorganisms, such as bacteria that form yogurt and cottage cheese, and, when necessary, by certain animal cells. Lactic acid fermentation has only one step. Pyruvic acid takes the hydrogen ions and electrons from the NADH produced during glycolysis, freeing the carrier molecule to be used again in glycolysis to produce ATP. The lactic acid gives yogurt and cheeses their tart taste. Muscle cells are one type of cell that can perform either aerobic or anaerobic respiration. During normal activity, muscle cells obtain energy through the aerobic pathway. However, during prolonged physical exertion or very strenuous activity such as running at top speed, aerobic respiration cannot supply energy rapidly enough for muscles. During that period of intense need, the muscles switch over to lactic acid fermentation. The lactic acid will accumulate in the muscle cells and will eventually be removed by the bloodstream. If the lactic acid continues to accumulate in the muscle cells, it can cause pain and cramping of the muscles. Fermentation energy. Output: fermentation supplies no ATP energy beyond that obtained from glycolysis. Cellular fermentation, therefore, produces a net gain of only 2 ATP molecules per glucose molecule.